and good morning. Welcome to NEUR3301. This is the Genetics of Mental Health. I'm your host, Harry McKay. I'm here to teach you about the genetics of mental health and various other topics. So thanks to everyone for joining me on the live stream this morning. I'm trying a little bit of a different thing this year, and I hope you all appreciate it. I've taught a couple of courses at Carleton since the end of the world, and they've been in various formats. The first one I taught, I did synchronous sessions over Zoom, and I did the whole thing where you ask all the students to turn on their cameras so you can pretend like you're in the room talking to people. That was a very tiny class. That was like a dozen people, so that wasn't maybe too much to ask. And then over the summer, I taught 1203. And there, just because of the sheer time constraints, I had to resort to pre-recorded lectures that students could watch at their own pace. And I think talking to students and looking at the various Reddits and comment boards where people talk about their experience of student life in the COVID age, what I've noticed is that the motivation to come to class is still always a challenge for people. It's maybe a particularly big challenge when you don't have any class to go to. You just have to make time to watch videos. But one of the main recommendations I've heard, and one of the main things I would personally recommend to anybody who's listening, is that if you schedule time and view the courses at a set time every week, typically the time the course is scheduled, that's the best way to keep yourself on task and actually stay engaged throughout the whole term. And for that reason, why not just host the class as a live stream? Then you get the best of both worlds. You can tune in at the scheduled time, and I'll be here giving you the lecture in real time. Or if you want to rewatch the lecture, or if you can't make it, or if there's some other kind of issue that prevents you from visiting the live stream, then you can come and watch it later on YouTube. And I chose YouTube as my platform. I don't know how many professors at Carleton are doing this, but YouTube is I think probably the most accessible platform out there. It certainly works better than the ones that Carleton provides, and you get the most flexibility, and you get the massive power of Google's $1 billion servers that enable high connection speeds, and it enables us to record the lecture and keep it hosted there. And of course, everyone has the YouTube app on their phone or their tablet and whatnot, so I think it's probably the most accessible way to do it. So that's what I'm going to do, and the videos will be hosted here on my channel, unlisted, so that they can't be found by the average person, but freely available to all of you in the class anytime you'd like to see it. YouTube should also supply closed captioning for added accessibility, and if you're one of those weirdos who likes to speed up your videos to 2x speed so that you can get through my lecture more quickly, that's of course an option when you're viewing the playback. So hopefully everyone enjoys this. Like I said, I think what I've heard from students and what my own experience personally would dictate is that you really are best off if you actually make time to come to the live streams and sort of set aside the hour and a half every morning to do this. Keeping yourself on task and feeling like it's kind of you're engaged with the class is a real big part of what makes this education worthwhile, right? I mean, you would scarcely need to be paying so much just to watch pre-recorded videos. I think coming to the live stream is superior for that reason, and it keeps you on task. And then the other good thing about it is that you can ask questions. So you can use the chat box in this in the YouTube app or the YouTube website to type in questions. You have to have an account on YouTube, which is one downside, but if you happen to already have one, that's probably ideal. If you maintain any subscriptions or whatever, you probably already have one. You don't need to use your real name. You just use whatever your username is. I'm not going to be too strict about attendance or anything like that. If your name is like, Weedlord420, might be embarrassing, but I'll probably never know who you actually are. So it's no big deal. So yeah, that's kind of the reason I'm running the class this way. Let me know how you like it. Hopefully this will work for everyone and give everyone kind of the best of both worlds, and lets me play with sort of fancier streaming technology. Anyway, so without any further ado, I'm going to get started going through today's lecture. And we're going to mainly just focus on sort of looking over the syllabus, going through a brief review or sort of introduction to genetics, and then we'll I'll get out of your hair, and then we can move on to more interesting and more neuro-specific topics. Oh, yes. So far, somebody already likes it. That's good. Um, okay, so let's jump into the lecture here. So this is the title slide. So I guess I'll introduce myself a little bit first. You may or may not know me. I'm not I'm not exactly present at Carleton all the time. I'm a contract instructor, so I'm sort of the lower class of the Carleton people. But my name's Harry McKay. Here I am at Carleton. I'm a contract instructor. You can, if you're trying to address me, feel free to call me Harry or Dr. McKay if you feel fancy. I think, at some level, it's probably less awkward if you call me Dr. McKay, because then you can never go wrong with being more formal. But personally, I would be fine with just Harry if you want to be casual. Doesn't make a difference really. So I did my PhD at Carleton I finished it back in 2015 in Alfonso Abizade's lab. So what I was doing in those days was studying the effect of early life exposure to endocrine disruptors, bisphenol A. If you remember, a while ago, people were really worried about the plastic that was in Nalgene bottles and lining tin cans, or cans and whatnot. That was BPA, and BPA does have some level of endocrine disrupting function. It acts a bit like an estrogen and a few other hormones. And what I was interested in was how this impacts the development of the hypothalamus, and in particular the feeding circuits of the hypothalamus that regulate energy balance. So I was looking at that from kind of a neurodevelopmental, neuroanatomical perspective, and it was all happening here in the former Life Sciences Research Building, the LSRB. This was a kind of an old, crusty building that was on the Carleton campus where all of the neuroscience research used to happen. I would imagine that all of you have never seen this building. It's now kind of clad in bronze, sort of looks like a lower level Trump Tower. It's along the I guess what is it? I can't remember the name of the street, but anyway, it's right in front of the river, and that's where that all went down. And that building was demolished after I left, and replaced by this, or not really demolished, but replaced by this new building that does something that nobody really knows what it does. And then right now, what I'm doing, which is kind of funny, is I'm working as a postdoctoral fellow in the Waterland Lab at Baylor College of Medicine in Houston. So I'm broadcasting to you live from Ottawa, but my actual job is in Houston. And I joined that lab back in 2015, and it's an epigenetics lab. So there I am studying the role of DNA methylation in brain development, and in particular the hypothalamus, and looking at how we can use novel approaches to analyzing whole genome DNA methylation data in a way that can help us understand brain development, the developmental origins of health and disease, and also just the way to use sort of peripheral methylation data to understand the to be able to predict individuals' health and disease risk later on. And that's all happening here at the Children's Nutrition Research Center in Houston. This is a USDA Texas Children's Hospital / Baylor College of Medicine building. Anyway, so I joined in 2015, and I was working there until basically the beginning of 2020, when, for reasons completely unrelated to the coronavirus, I moved back to Ottawa and was going to finish my postdoc remotely. But of course, the world ended, and I was supposed to be traveling back and forth on a monthly basis to do lab work and so forth, but of course that became impossible. I've only actually been back a few times since then, but I've been working remotely on the kind of the bioinformatic analysis of all these data, and also just writing the paper. So I'm kind of, in a sense, I'm lucky to have a job, but in another sense, it would be really nice to have a job here in Canada, other than this one, which is just sort of a side hustle more than anything. But my interest is in, genetics and epigenetics and how they relate to the brain and disease risk and mental health. So that's why I'm here teaching you this course. I've taught a few courses at Carleton before. Back when I was a PhD student, I was teaching the 1201, which was sort of the precursor to 1203 and 1202. Then in the winter of 2021, I taught 4001, which is a special topics class about some of the same kind of stuff here. And then I taught 1203 over the summer, which was quite a handful, I have to say. Summer courses are very tough as students, but they're also fairly difficult to run as instructors. Anyway, so that's all about me. You may not be too curious about that, but I think it's always a nice gesture to give a little bit of detail about my own background and career. In case you are curious, I can tell you more about it if you ever wonder how I managed to get where I am today and the kind of pathway I took. Okay, so now we're going to review the syllabus, and I want to take some time and just look through what this course is really going to be about and what I'm going to ask of you and all that good stuff. So let's jump into the view of the syllabus here. So you can find this on the Brightspace page, and if you haven't taken a course yet since the summer, Brightspace might be a bit new to you. It certainly was new to me when I taught 1203 this summer, and that was a source of many problems for both me and the students. But it's not that bad, and I think I've gotten a bit better at laying the course out in a way that people can understand. So take a look at the Brightspace page. It's divided up into modules. I haven't added most of the modules yet because I haven't actually made most of the content for this course yet. It's being prepared dynamically as I go. So all I have to do is meet the deadlines. But anyway, the course is divided into modules, and the first module is just a place containing the syllabus and the course textbook. So here's the syllabus. This is 3301. I'm Dr. Harry McKay, your friend and instructor. Your TAs are Sarah Siddiqui and Priya Van Oosterhout. Their emails are here. If you visit the groups function on Brightspace, you'll see which TA you've been assigned to. They're assigned randomly. The class is simply divided in half, and you're assigned on a completely random basis to one of our 2 TAs. The TAs are also watching these lectures and working on the assignments, so they should be very well qualified to help you with any problems you're having when it comes to the technical side of things on the assignments. We'll get into that. So what we're aiming to do in this course is recognizing that mental and neurological diseases have genetic and epigenetic components. We want to understand how those work. Now, some of them are like Huntington's disease, where there's really a single mutation that determines whether or not you have the disease. That's reasonably simple to understand. Others, like schizophrenia, really seem to run in families and clearly have some kind of a genetic component. You can tell from twin studies and the like, and pedigree studies. But you really can't tie them to a single mutation in most cases. The latter, I think, tend to be the more interesting and tend to be more the target of modern-day genomics studies, because you really have to take a whole genome approach to understanding how these, where the risk for these diseases come from. So these modern approaches of genomics and epigenomics are enabling researchers to look through the entire genome and try to identify disease-associated mutations, variants, and epigenetic marks and so forth. And so what we're in the middle of right now is sort of a gold rush of information. More genomic and epigenomic information is being released on a weekly basis than you could have even begun to fathom. When I started university, it was 2004, and relative to now, the information that was available in those days was just absolutely microscopic. It's gotten to a wildly advanced stage now. So we're going to start to cover some of the basics of heritability, genetics, and epigenetics, and then the methods that we use to study them. And then the good part of the course starts, which is when we actually go through individual conditions and phenotypes and learn about how the studies that exist today have taught us the genetics and epigenetics of those things. So we'll look at things like schizophrenia, autism spectrum, addiction, a whole bunch of other good stuff. And at the same time, I have a goal of teaching you how to work with the data yourself a little bit, how to use some of the online tools to work with some of the data sets, just in a way partly to expand your skill set, but then also partly to help you understand where these data come from and what it is the researchers are doing, so that if you see a figure in a paper, you'll know how it was made. So I guess I have a mistake here already. There are prerequisites. The genetics courses in the bio department, I believe, are prerequisites. And with that in mind, I am expecting or hoping that you all have a reasonably high level of understanding of kind of classical genetics, things like, how mitosis and meiosis work, and kind of classic Mendelian patterns of inheritance and all the fruit fly stuff. I am kind of hoping that you're on board with that already, because I want to kind of just jump straight into the brain stuff and the modern genomics as much as possible without reviewing too many of the kind of classic topics. So what I want to achieve by the end of this course is I want you to understand the basics, of course. I want you to understand some of the methods, including bioinformatics to a limited extent. I want you to understand how genome-wide association studies, or GWAS studies, work. They're the main kind of genre of study we'll be talking about here. That's now in the modern kind of days, this is how most of the connections between genotype and disease risk are made. I want you to understand the big data approaches to genomics and epigenomics. I want you to learn how to interpret figures like heat maps and principal component analyses and correlograms, and these kind of more flashy advanced figures that you tend to find in these kinds of papers. So it's very much also a course where I'm trying to teach you how to interpret data. We want you to use several web-based tools to explore the genome and so forth. And then also I want to make sure you understand the limits of this technology and the challenges that exist in trying to make it equitable and universal. So that's kind of the topic of the last class I have planned, but it's an ongoing theme in the whole semester. Now, let's get into readings. I don't ask for you to buy a textbook. I think if you happen to have a genetics textbook from one of your past classes, that would be a great resource to keep on hand. But I really don't like the academic textbook publishing industry. I think it's pretty evil. So I would never want you to buy a book. If you can find a book that fell off a truck or use one you already own, or maybe buy one used from Haven Books or somewhere, that's all fine. The book that I'm assigning, quote unquote, is called Online Open Genetics, and it's just a free online textbook. The address for it is given here in the syllabus, but there's also a PDF copy of it posted on the Brightspace page. It's fairly bare bones, but it's really only meant as a optional supplement to give you some of the starting points that you need to understand this course if you need to brush up on your genetics a little bit. Lectures are streamed over YouTube, as I said. The recordings will be available within 24 hours. I think they're actually going to be available instantly because YouTube DVRs them and posts them almost immediately. So you'll be able to follow the same link and find this recording when you want to watch. Attendance, of course, is not mandatory, but I would really hope that you choose to attend as many as you can. I think that's just a better practice for your overall learning and even frankly for your mental health. Having a routine, I guess it depends on what kind of person you are, but for me, having a routine is like essential for maintaining a stable level of mental health. If my routine is off kilter, then I'm not going to be a happy guy. So, your mileage may vary, but I would highly recommend committing to coming to the live streams and trying to engage in that way. The lecture slides are posted about 24 hours after the stream. You might be asking, how come I don't post them before? The reason is that I often find mistakes in my slides and when I'm going through the lecture, and it's really annoying to have to replace them on Brightspace, and then students might have the wrong version downloaded already. So I post them after I've had some time to reflect on them and fix any problems that they might have. I always note, and you folks are all, I guess third year or higher, so I don't need to really tell you this, but you can't just have the slides and call those your notes. You really ought to be taking notes either by hand or on your computer. I mean, I still take notes by hand every day. I have one of these books that I fill up, and it's still something I do. I find writing them out by hand is a little bit better, but again, your mileage may vary. You're at this point in your careers, you probably know better than I do how you like to learn. There's discussion forums on the Brightspace page. I just have kind of one all-purpose forum, so if you have any questions that you feel like asking publicly, either about the course or about the assignments or about just kind of genetics in general, feel free to post them up there. Other people might enjoy jumping into the discussion. Office hours are going to be held by Zoom, so I'm scheduling them for Thursday from 1 to 2 p.m. I'll send out the link every week to remind you how to view it, but it's just office hours. You can hop in and hop out as needed if you have any questions, or if you want to just kind of hang out and see what other people are talking about. If nobody's talking, I'll probably just riff for an hour and just kind of give you a stream of consciousness. There's no problem there. You don't need to have the camera on or anything. You just come in and use the chat window or ask over the microphone, or you can have your camera on, whatever you like. If you need to talk and you can't make the regular scheduled time, just email me and we'll try to make an appointment. TAs, you're randomly assigned to your TA, so it's not alphabetically, it's just random. You can see that on the groups page. Communication, best way to contact me is by email. Of course, if it's anything private, that's the only real way to do it. You can ask over the forum too, but I tend to be quicker on the emails than I do on the forums, so if it's an urgent question, email is the best way to reach me. Or you can visit my office hours. Okay, now here's the actual stuff the course is made of. There are... I'll start with the two midterms. So we've got two midterms. They total 40%. They're worth 20% each. The first one is October 4th. The second one is November 15th. They cover all of the content from, the last midterm up until the current midterm. So they cover kind of everything that came before them. They're going to be held on Brightspace. They'll be in the quizzes section of the Brightspace page once I make them. I found that it's fine to leave them open for 24 hours. So they'll open up at 8 a.m. the day of the, say October 4th, and they'll close at 8 a.m. the next day. And you have 24 hours where you can hop in, write the exam. The exam is going to take about an hour and a half. So you can just hop in and write it whenever you think is best for you. And then submit, and we'll get more details on that as we come closer. But it's likely going to be a mixture of multiple choice and short answer questions for both the midterms. The final exam is TBA. I have to decide what I want to do for that, but it'll probably also be online, and it'll probably be pretty similar to the midterms. I found that the online exams actually seem to work reasonably well in terms of getting people grades and getting them, testing their comprehension. You actually, even though it's online and it's technically sort of open book, you really can tell who understands stuff and who doesn't. And then the assignments, there's 3 of these, and they're worth 30% in total, 10% each. And these are kind of like web quests or sort of hands-on learning activities where I'm going to post a video. And the first assignment is already up, by the way. I post a video where I walk you through how to use a web-based tool like the UCSC genome browser to explore the genome. This is a tool that real researchers use every day. And then I give you a few challenging, a few questions about that test your ability to kind of follow through the steps I did. So, find this gene, how long is this gene, how many exons does it have, all this kind of stuff. So this is kind of an experimental thing for me. I haven't had these assignments before. I'm just kind of design them here for this class. So we'll see how they go, but I think it should be fairly straightforward. The first one is an introduction to the UCSC genome browser. It's due October 6th. The second one is how to analyze RNA-seq data and gene sets. So that's November 8th. And then finally, genomic ranges and epigenomic data, December 8th. So that's at the very end of class. So I would recommend, so right now what's happening is I have the TAs experimenting with these assignments and doing them themselves. And the first thing I'd like to know is how well it works for them. Based on that, they'll learn how to do them themselves. And that's when you need help, you'll be able to email your TA for help on these assignments. So I would wait a week or two before trying to start them, just to get some more of the class under your belt before you go, and to let the TAs get a chance to learn how to do these assignments themselves. So yeah, we'll see. I think that's going to be pretty interesting. It'll be kind of a lab activity, and when you're finished, you'll be able to tell, future employers or future labs that you might want to join, or whoever or wherever you're headed, that you do know how to do a little bit of basic bioinformatics. And that's a good starting point, because once you start to learn a little bit, it becomes easier and easier to learn more. So here's the outline. I don't want to go through everything here, but basically this just gives the dates of all the different topics. Today we're on September 8th, the course Introduction and Basics of Genetics. We have a lot of sections of the textbook for you today. And then, here and there, I've given you a few other bits of the textbook that I'd like you to take a look at, before or around the lecture. But for the most part, the textbook doesn't become, the textbook kind of, we don't really tend to use the textbook a whole lot after the first few lectures. And the assignment due dates are given here with these superscripted notes in case you forget. Okay, so I don't want to spend too much more time on the syllabus. It's all very boilerplate stuff. You guys, you folks have seen many a syllabus in your day. The course uses Brightspace and Zoom and Carleton. So this is also out of date. It's not Carleton's MediaSpace or Zoom now. It's YouTube. So I'll maybe try to change that. See, this is why I don't post. I don't like posting lectures before before I've gone through them. So there's a few comments here on the difference between Brightspace and CU Learn. This could definitely cause problems for people if you're not used to the new platform. Carleton has a lot of tutorials on it, and it is a bit of a learning curve, but that's just part of life. You got to learn the topic, and you also have to learn the weird web interface. I will point out the syllabus is the ultimate source of deadlines. So sometimes Brightspace will show things weirdly, or maybe not show a deadline, or maybe not remind you enough. I've definitely seen that in the past. You're in charge of reading the syllabus and making your own calendar entries based on the due dates here. The syllabus is the ultimate source of due dates. Nobody else, Brightspace is second to the syllabus. So the syllabus is gold. Yeah, I mean, so just read through these and make sure that you understand all of them. Uh, Oh, assignments are going to be submitted in Microsoft Office format. This is written on the assignment, but I'll just stress. So either doc or PowerPoint or Excel files, depending on what's appropriate. This is kind of all purpose. You can also submit as plain text in either text or comma separated values if you like, or PDF. PDF is a great way to submit because you're guaranteed to see the exact same thing on every screen. I just, we had in 1203 this summer, people were sending in stuff from like what's the Apple program pages, and the TAs couldn't open them, and then that's a big thing because it's like, well, if you send something in on the deadline but it's in a format we can't open, what do we do? Do we charge you a late fee or do we not? That's always a problem. Oh, yeah, speaking of late fees, let me describe the late penalties for the assignments. So they're always due at 11:59, like a minute before midnight on the due date. This is another funny thing. Like I used to make the due dates at midnight, and students got very confused by what midnight. I mean, it's understandable actually, but 12 a.m. means the very first minute of that day. So if something's due at 12 a.m. on September 8th, then right now you'd already be like 9 hours late. But a lot of people thought that 12 a.m. meant midnight on September 8th, like so at tonight. Anyway, so 11:59 on the due date. The late penalties are 10 percentage points per day. So the assignment is worth 10% of your grade. 10 percentage points out of that would be basically a drop of 1%. So if you are 5 days late, for example, even if your assignment is perfect, the most value it could contribute to your grade is 50% of what it normally would. So you would get, essentially, if your assignment is perfect but it's 5 days late, you would get 5 out of 5. That's still, 5 out of 10. So you can only get half the value for that assignment if it's late. And so that's quite a steep penalty. If you have problems, if there's a medical or personal reason why you can't get your due date on time, or if there's even by all means email me. I don't want to be some kind of I don't want to be over the top with how strict this is, but the due dates are firm in absence of some other kind of mitigating factor. So please let me know if you have any questions about this. Otherwise, we'll jump back into the lecture here. So we have to ask, and this is the basics of genetics, so I'm only giving you kind of a few highlights, maybe a bit of a highlight reel of the things in genetics that I think are going to be especially important to the course that we are going to be talking about. It's not all-inclusive by any stretch, at least at this stage. So humans have always been aware that individual characteristics are passed down to offspring. This has been noted in livestock and agriculture, of course. Selective breeding was going on long before anyone had any clue what genetics was. We have records of domesticated species dating back thousands and thousands of years, long before Mendel or anybody else like that. And of course, this is known in humans as well. There's a fixation on bloodlines as a biological justification for preserving wealth and power. If you feel that your bloodline is special or has some kind of intrinsic value, the motivation to keep that bloodline pure and marry within it becomes stronger and stronger. So this leads to things like the Habsburg dynasty. These were a bunch of heavily inbred Europeans who ruled much of Europe through the early part of the last millennium. So Charles II of Spain, for example, he has this famous Habsburg jaw that resulted from many, many generations of inbreeding between first cousins. Ultimately, these people ended up so diseased and so impaired by this kind of inbreeding that they were essentially non-functioning as human beings. But the fixation on bloodlines as a means of preserving wealth and power still remain. Of course, inheritance works along bloodlines, and your mom or dad can get you a job at the dealership and get you started and whatnot. It's kind of the same sort of thing even today. But this is all known long before genetics was ever a glimmer in Mendel's eye. Um, the while it was known for most of history, or like understood for most of history, it had no real biological basis. Even Gregor Mendel's studies didn't have biology really as a biological basis as a main feature. They were simply analyses of traits, and you don't need any biology to understand that. So Mendelian inheritance, if you haven't looked at this in a while, I would recommend boning up on it a little bit, because it is a kind of a guiding principle in the overall field of genetics. Prior to Gregor Mendel's work, inheritance was sort of assumed to be blended. And so if you imagine, say, for example, a phenotype like color, say flower color here, and you've got a purple parent and a white parent, the theory of blending would hold that the offspring of these two, when you were to mate them, would just be kind of a 50-50 mixture of both, kind of a light purple, I guess. And this is actually not terribly, this kind of makes sense in an intuitive way if you look at how human offspring or human breeding tends to work. If you have a tall parent and a short parent, typically the offspring of the two is somewhere in the middle. That's actually more regression to the mean than a property of genetics. But still, this is sort of intuitively what you think. And the same would go for skin color and other kinds of continuous phenotypic variables. So this logic was applied to most forms of inheritance. And the big innovation was, at least from the part of Mendel, was falsifying this concept of inheritance. It would be impossible, theoretically, if inheritance were blended, for you to take a blended organism and then separate out the constitutive traits after they'd already been created. And like, in a way, I suppose it's a little bit like our understanding of color and light prior to Newton, because Newton was the one who showed that you could take a prism and separate white light into its constituent spectrum. Spectrum being a word, Latin word meaning ghost, so it was kind of like a ghostly appearance that the rainbow would cause. But anyway, showed that like white light was actually a combination of separate constituent wavelengths. And in the same way, Mendel showed that the phenotype is actually constituted of a variety of separate contributing genes that can be combined and re-separated out again. So his biggest achievement was falsifying this concept of blended inheritance. Discrete traits like P, shape, and color are never blended. It's either one or the other. And if you breed them carefully, you can show that some traits are dominant, meaning that if there's any trace of that trait in the organism, that trait will take over. And others are recessive, meaning that they can be hidden away, latent, and then re-emerge after breeding in a certain way to remove the competing dominant alleles. And this is it really can't be overstated how innovative and how important this finding was. However, most of the things we're interested in here in the genetics of mental health don't really follow these simple rules of Mendelian inheritance. Most are polygenic. Mendelian inheritance is a guiding principle because on the level of any individual allele, the principles that Mendel discovered apply 100%. This is how inheritance works at the level of individual alleles. The difference really is that most complicated phenotypes are not the product of one single gene or the presence or absence of one single allele. They're the product of many, many alleles and many, many genes all interacting and working together. That's polygenic, and that's how most things actually work. So while you can follow this for individual genes and trace the history of individual alleles in this way, the way the conditions we're interested in work, they're more polygenic. This, by the way, is the reason what we're looking at genomics on a genome-wide level, because we need to understand what's happening across the genome to make sense of these complicated traits like schizophrenia or personality and that type of thing. So Mendel's first law basically posits that, the law is called the law of equal segregation. And this is really the reason that anything works at all in genetics is because, in sexual reproduction at least, gametes contain one or the other allele. They do not contain both. If gametes contained both alleles, that you have two different alleles for every gene, right? You're sort of a diploid genome. If gametes contained both alleles, then it would be impossible. You would essentially just be a clone of your children would be a clone of you. You just pass along all the same alleles you have, and your child would be an exact replica of you. And that would really not allow any kind of evolution or breeding or mixture of traits to take place, and it would actually kind of make sex as a concept pretty redundant. Traits exist in different versions called alleles. The word allele comes from a Greek word meaning other. You'll see this prefix al fairly often. What's it called? Allotypes, I guess, and allometrics. That's the other one. Allometrics are when you measure the size of things in different species and scale them to be relative to other things. So you can talk about like brain size as a function of body size and so forth. So it just means other. So if both alleles of a given gene are the same, you can say that the organism is homozygous for that gene. And if the organism has one of each, it's said to be heterozygous. Very simple stuff. And bear in mind that these are all, the work that Mendel was doing concerned dichotomous traits. Now, alleles in a diploid organism are dichotomous. You have one or the other for the most part, but the traits they give rise to, again, most traits being polygenic, are not dichotomous. They're kind of a combinatorial sort of thing. Now, something that's important to understand about kind of the classic way that genetics was studied was, if you kind of take Mendel's work as a template and you think, how could I apply this to other things where you don't have the short life cycle of the pea plant? You can breed them and pick out traits and breed them again. That's something you can do with a model organism like a plant or maybe a Drosophila or a fruit fly or an Arabidopsis plant or something like that. But for human beings and for livestock and for more complicated kinds of creatures, that type of method is really not possible. However, you can look at what nature, you can look at what happened naturally and try to analyze on the basis of that what went down. So the word pedigree, you can look at the family tree more or less and follow traits through the family tree. This is a pedigree analysis. The word pedigree comes from Old French pied de grue, which means like a bird's footprint. If you look at the kind of family tree that we'll see in pedigrees, and we will look at some pedigrees down the road, it kind of looks like this sort of thing. And I guess in somebody's mind, that looks like a bird's foot. I think a bird's foot kind of looks more like, like a bird's footprint kind of looks more like that. But, I guess if you use your imagination, that's believable. So this is a key method in classical genetics in order to use to infer the inheritance pattern of certain traits. And there are some traits where, like, if you look at the pedigree, it becomes blindingly obvious how it's inherited. If it's X-linked, or if it's a recessive or a dominant allele, you can tell a lot of these things based on the pedigree chart. And the symbols used are given here. So if for any given trait, let's say it's a disease, like say for example Rett syndrome, you can, Rett syndrome is actually kind of a bad example, but anyway, say it's Rett syndrome because I just thought of that. You can look at individuals as either affected or unaffected, meaning they either have the syndrome and they're colored in black, or they don't have the syndrome and they're colored in white. The sex of the individual is given by the shape, so it could either be female, meaning circle, or male, meaning square. Technically, this is speaking about chromosomal sex, which is the most important thing when we're talking about patterns of inheritance. You can give a diamond if you don't know the sex, which is kind of like a square turned 45 degrees. You connect them using these lines. So siblings are connected with kind of a branched line. Mating that is given by a straight line between the two different shapes. And consanguineous mating, which means Latin for of the same blood. Sang means blood, of course. Con means with. it's involving second cousins or closer. So our friends, the Habsburgs, would have had a lot of these consanguineous matings. So you can keep track of all this and follow the pedigree through, and figure out where the disease alleles might be coming from in the history. And this actually enables you to determine a number of modes of inheritance. Some of these, I think, probably a lot of these we actually will talk about at various times in the class, so you can have this as a reference. But you've got your classic autosomal dominant. Autosomes are any chromosome that's not a sex chromosome, so chromosomes 1 through 22 would be autosomes. And a dominant trait, of course, is one where if you have the allele, you're going to express that trait. So it's a trait that's caused by a dominant allele on an autosome. There's no sex bias in these because, of course, both biological sexes have the same autosomes, and there would typically not be any difference between the likelihood of a male getting this condition or a female. These also tend to appear in every generation because odds are pretty good that one of these dominant alleles will make it down to the next generation. It's a 50-50 coin flip, so if you have 2 kids, the odds are reasonably good that you're going to get at least one of them that has this trait, if not both. There's also X-linked dominant. This is when the dominant allele causing a disease or a trait is on the X chromosome. Of course, both males and females have an X chromosome, but females have 2, not 1. So typically, on X dominant ones, you see a higher frequency among females, because basically, if you're a parent to say having one, you're a parent with one. Say you're a mom, I guess, for example, you've got one good X chromosome and one bad X chromosome. The odds are 50-50 that you pass it along, and so typically this is going to appear in females more often. Also importantly, it's impossible for fathers to pass this trait along to their biologically male sons, because fathers, in the case of sons, dads can only pass on their Y chromosome. It's impossible for a dad to pass a X chromosome on to a child and have that child be biologically male, or chromosomally male, I guess we could say. There's also autosomal recessive and X-linked recessive. These are when you need 2 copies of the allele present to express the phenotype. So what you'll see here is this phenotype will skip generations, and it takes many people in the phenotype are just carriers. But if you look at where it's expressed upstream, in the grandparents or the great-grandparents, and you follow it down, you can typically guess which people are carriers, because you can almost follow the allele through the family tree and figure out what path it must have traveled to reach the proband. The proband, by the way, is whoever it is you're looking at the end of the family tree that has the condition you're interested in. And the same sort of thing is also true for X chromosomes. You also have Y-linked inheritance. The Y chromosome is kind of a stubby little, kind of a little stump of a chromosome. It doesn't have a lot going on there. But it does exist, and of course, biological males have it. It can only affect males, and it can only be passed on from your father. There's not too many of these because the Y chromosome, like I said, is not very gene rich. It's not very big just physically in its size, but there's also not a huge number of genes on it. So there's not a lot of room for mistakes to crop up. And then finally, there's mitochondrial inheritance. We aren't really going to be talking about this, but there is also mitochondrial DNA. The mitochondria, of course, are believed to have been former separate organisms that invaded the prototypical, eukaryotic cells and established a little partnership. So the mitochondria actually do have their own DNA, and that DNA can also have problems with it. You can only inherit that from your mother because your mitochondria come from your mom. Now let's look at population genetics. The population genetics is sort of the study of variants and their relative proportion in the population. So this is, I think, fairly relevant to what we're studying, because we're looking at things and we're trying to understand how genetic variation contributes to disease risk. And diseases have a certain proportion or frequency in the population. Schizophrenia, for example, the prevalence is about 1%. You have to kind of have some way of squaring the prevalence of the disease with the prevalence of the genes you think are causing it. If you have extremely common variants or alleles, it's unlikely that they contribute to a disease very much, because if they're extremely common, it would seem to follow that everyone would have that disease. And that's clearly not the case for most things. So we want to know about the frequency of these and use that to infer how often we might see them combined together in a way that gives rise to a trait or a phenotype that we're curious about. So really what we're asking then is how common a given allele is in a population. This has one important offshoot when you're thinking about how to design these studies and ask these questions that I think almost forms kind of an epistemological limit to what we can learn through genomics. It's very difficult to understand the relationship between a rare allele and a very common disorder. The reason is this. To do a genome-wide study, you need many, many participants because there's a lot of genetic variation. Most of it is just noise. Very little of it would have anything to do with the trait you're interested in. And you need a large sample size to kind of compensate for that. Imagine you have a rare allele that makes up 0.001% of the population. It's an extremely rare allele, so one in 100 or no, 1 in 1,000 people or something has this allele. Very rare to find it. Imagine if you're trying to determine if that allele is causing, say, heart disease or schizophrenia also. Just to keep with the same theme. What you would need to do is basically show that the people who have this allele are more likely to have a condition than the people who don't have the allele, right? Now, think about what that would be like given such a rare allele. Schizophrenia makes up about 1% of the population. So you'd have to, just to start with, you'd have to sample 100 people to even find one person who has schizophrenia. So you sample, let's say, 1,000 people, and that's a lot, right? And then, 1,000 people, and that's a lot. You would have about 10 of them that have schizophrenia. Now imagine if you're genotyping those people with schizophrenia, and 6 of them have this allele, and 5 of them don't. I'm kind of confusing this all. I should just stick to the slide. But anyway, the point of the matter is that even if you genotype thousands of people, you'd only get a few hundred that even have this allele. And it's very difficult, given that, to make statistical associations between rare alleles and diseases. Ultimately, then, to understand the allelic structure of the population you're studying is essential in making sense of the contribution of different alleles to disease risk. Let's just leave it at that. I think it's better to just say that than try to improvise an example on the fly here. But I think you can kind of understand this. It's difficult to link rare things in a statistically robust way because they're so rare. You almost don't have enough people to even make kind of a to have a good understanding of what's happening at all and to separate signal from noise. So allele frequency is a fairly relevant thing here. You can determine this empirically simply by genotyping a large sample of the population. So for diploid organisms like us, where there's just 2 alleles, there's, say A, capital A, and lowercase a, all you have to do is genotype a lot of people and count the number of times a given allele appears divided by the sample size, and that is the allele frequency. Typically, when you're looking at this kind of stuff, there's a major allele and a minor allele. The major allele being the more common allele in the population and the minor one being the less common. Sometimes you'll also have multiple minor alleles where the major allele makes up, say, 75% of the people, and the remaining 25% are made up of 2 other kinds of alternate alleles. That's also possible. This has to be determined on a population-wide level using fairly careful genotyping. You have to get a lot of people and look through their entire genome and figure out what variants they have, how common those variants are in that population. And then, of course, it varies between populations. So what the allele frequency that might be true, say, among white Europeans might be completely different from people who are indigenous Taiwanese, for example, or anybody else who you could name on the Earth. The Earth has a very large and fairly stratified population structure. So the allele frequency can differ quite substantially between different groups. Now, in the absence of natural selection, the Hardy-Weinberg principle, which is quite an interesting kind of mathematical contrivance, states that allele frequencies should remain constant across generations. So this is really, I think the way to understand that is to contrast it with earlier thoughts or earlier ideas that held that dominant alleles would eventually become more and more frequent with time. People understood allele frequency reasonably early into the history of genetics, and I think if you kind of just reflect on it logically using kind of your own reasoning, you might come to believe that the dominant alleles would become more dominant with time, because that's just kind of how things work, right? Amazon.com started as a little bookstore online in the 90s, and it became bigger and more powerful until practically everything you buy now is on Amazon, or if not that, Walmart. And this is sort of the way things work. Things that have a lot of power and influence tend to become more and more powerful over time. They tend to monopolize, as you might say. And that's just sort of how nature appears to work. If you have a dominant species in a given ecosystem, theoretically, that species should become dominant to the point of eclipsing every other species. This happens, I guess, when you're competing in the same niche, but it obviously doesn't happen across niches. You know, a tiger can dominate a rabbit, but you can't have a world that's only tigers. You need some rabbits in there to feed the tigers, kind of thing. Anyway, this was a kind of a question that was sort of open. The Hardy-Weinberg principle answered this by showing that you can, you know, for a broad range of allele frequencies, if nothing is happening to naturally select one allele over the other, meaning that the alleles are completely neutral with respect to survival and reproduction, those allele frequencies will remain constant in perpetuity. So the model here kind of gives you a sense of what that means on a kind of continuous level. So on the x-axis here, we have the frequencies of the dominant and recessive allele in this case. The dominant one is the P allele, and the recessive is the Q allele. And of course, they're the only two alleles there are. So the proportion of the dominant one is always sort of the opposite of what the recessive one is. So if you have full, you know, if the dominant allele makes up 100% of the population, then of course 0 is left over for the recessive, or it could be 50-50. And then these lines are the proportion of the population that has a given genotype. So AA meaning both copies, they're homozygous for one allele. Capital A, lowercase a means they're heterozygous, and then capital A means they're heterozygous for the major dominant allele. And anyway, so if you're homozygous for the P allele, and everyone is like that, here's where you'll be. You'll be at 100% homozygous. If you're looking at everyone having the Q allele here, everyone will be homozygous, for the Q allele. But of course, that's not how things really work. What actually happens is that there's usually a mixture, and let's say that the P allele makes up, let's say, 70% of the population, and thus the Q allele makes up 30%. What you would expect there, in the absence of any selection, is that the frequency of the double A genotype would be a little bit above 40%. The the heterozygous phenotype would be about the same. And then the the homozygous phenotype for the minor allele, for the recessive allele, of course, the recessive allele is the lower frequency one, 30%. This would be lower. It'd be around maybe 10% of the population. So this just breaks down how you would expect the allele frequencies to kind of shake up, how the genotypes, I guess, you'd expect to shake up based on the allele frequencies in the population. And this is happening in the absence of natural selection. So what really matters is if you look at allele frequencies and find that this curve is like shifted in some way, so that the frequency, say, of homozygous alleles is a lot rarer than you would think. What that would imply is that the genes are actively being selected against, and they're changing their frequency over time. So it's a kind of an easy way to estimate how rapidly something is evolving in a population. So in the genomics age, which is the age we're talking about, allele frequencies can be determined for variants on a genome-wide level. So most variants don't really do anything, as far as we can tell, or whatever it is they do is fairly minor and doesn't have a huge effect. We actually don't tend to talk about dominant or recessive alleles very much in the world of modern genomics. I'll tell you when you need to know those terms for a given gene. I think, things like Fragile X or Huntington's, we can get into dominant and recessive stuff there. But for most of these conditions, it's not really the way we think about things anymore. In the genomics age, alleles are linked to conditions and traits more by like odds ratios. Like if you have this allele, your odds of developing schizophrenia are 50% higher than if you don't have the allele, that kind of thing. That's the way things are expressed more now, not dominant or recessive. Like I say, the modern world of genomics is a lot more complicated than what we can do with P plans and so forth. I'll point out, by the way, that in some cases, alleles might be in a Hardy-Weinberg equilibrium, meaning that there's no evidence of selection taking place in one population but not in another. This is kind of relevant when you start to break out and look at the worldwide, the world of genomics across the broader world. The majority of like association studies that try to link traits to genes are done in like white European descendants, just because that's where most of the money to do the research is located, and that's where most of the kind of experience is. But the kind of population genetic structure can vary a fair bit in different populations. So what a given allele may be or may not be selected against in one place, but it might be selected against in another place. So the 1,000 Genomes Project, which you'll hear about a fair bit in this class, was an attempt to build a global genome-wide atlas of human genetic variation, so that we can really understand how the human genome varies in more than just European white people, and use that as a basis for kind of learning more about the genetics of diseases. So they had a couple of different phases, phase 1 and phase 2, and this shows all the different populations they sampled from and where they were located. So you've got you've got Finnish people who sampled from Finland, and you've got British people sampled from Great Britain, and you've got, anyway, there's these acronyms, I forget what most of these actually stand for, but anyways, you've got people from all over the world at the very least, which is certainly a good place to start. Now, it probably doesn't need to be said, but DNA is the genetic material. This is what genes are actually made of. Mendelian genetics and indeed much of classical genetics really didn't have this as like a definitive concept for a lot of the time. It was studied, inheritance was studied a great deal before DNA was ever discovered. This is a little bit like how Ben Franklin would study electricity without ever knowing what an electron, much less an atom, even was. You can do a lot of work with natural phenomena without having any understanding of the underlying physical basis. This is one of the beautiful things about science, actually, is you can do a lot of experiments with just your mind if you kind of forget about worrying how reality works. But Mendel's theory worked very nicely, and it worked reliably without even knowing the physical basis. So maybe all the physics we know is actually kind of overrated. But a number of experiments in the 20th century did lead scientists to conclude that DNA must be the carrier of genetic information. In the first half of the 20th century, protein was a main competitor for people knew that the genetic information was stored in the cell, most likely in the nucleus. People knew that the nucleus basically had nucleic acid, and they knew that it had protein, the histones. And they sort of figured, well, nucleic acid seems very simple chemically. There's just 4 kind of ingredients to it, ACTG. There's no way that has the kind of information we need. Proteins, on the other hand, they knew were very, very complicated and could carry a great deal of information. So they must have thought, they really did think proteins must be carrying the genetics, and that's where the inheritance comes from. But a lot of these early experiments really show that it's actually DNA, not anything else, that could carry this information. You can, I would recommend looking into this a bit more in your own time, because a number of interesting experiments were done. But anyway, by the 1940s, people widely accepted that DNA must be the molecule of inheritance. But people didn't really know how it could carry genetic information. You can break it down into 4 types of nucleotide, adenine, cytosine, thymine, and guanine, ACTG. But that just kind of seems fairly simple. Now, Chargaff, who was a scientist, kind of a chemist, was breaking down DNA and quantifying the relative amounts of the different nucleic acids, and found that in a variety of species, A and T were present in the same proportions as each other, and G and C were present in the same proportions as each other, suggesting that there was some kind of link between those 2 pairs of nucleic acids. But anyways, it was a fairly simple molecule, and it just wasn't clear how it could carry information. nobody knew about what its three-dimensional structure was, so it was impossible to to figure out what, I mean, if you take A, C, T, and G and just mix them up, that doesn't tell you anything. If they're in a particular order and in a particular structure, that would tell you something, but people had no idea what the structure was. So the three-dimensional structure was ultimately credited to Watson and Crick, as well as Maurice Wilkins and Rosalind Franklin, who were working at, I guess, Cambridge at the time. So they did the X-ray crystallography. If you ever want to read The Double Helix, written by Watson, I believe, that's quite a nice book about the early history of it, but there's been countless books written on it and that have tried to kind of revise the history and look at what really happened, because Watson especially is not a particularly nice guy. Crick is dead now, but he was much nicer. But anyway, famously, the two relied on an X-ray diffraction photograph stolen from Rosalind Franklin's graduate student, Raymond Gosling. He was the one who actually took the photo, working in Franklin's lab. The photo was called Photo 51. It took a lot of skill to crystallize these bits of DNA and actually get an X-ray photograph of them that could be used. So, once you have the photo, you can do the math and figure out what the structure might be. But getting the photo itself is technically quite challenging. So that was stolen or borrowed, I guess, probably stolen from Franklin's lab and her grad student. Photo 51, I took this picture when I was in the Wellcome Trust Genomics Centre in Hingston, UK, which is right outside of Cambridge. This is a big conference centre and research lab centered around genomics. The UK, despite being kind of a failed empire, is still a world leader in genomics and genetics. Oftentimes the prizes last a lot longer than the great fame and the great success. But anyway, this is a wonderful center, and they've got Rosalind Franklin Pavilion here where you can go and hear talks about genomics. So she did eventually get some recognition. The paper that Crick and Watson wrote has this great line in it. It has not escaped our notice that the specific pairing we have postulated, that's like ATCG, the base pairing, immediately suggests a possible copying mechanism for the genetic material. So this it has not escaped our notice. If you go on Google Scholar or PubMed and just type that in, you'll find all kinds of cranks who have copied that for their own stupid discoveries of I discovered a new kind of a new type of behavior that whatever. I mean, just people will overuse that term as though they're making such an equivalent discovery. Anyways, I don't want, not to editorialize too much, but it was really pretty much a earth-shattering world historic event to discover the three-dimensional structure of DNA, and so all the people who were involved deserve a great deal of credit. Although I think, to be fair, Linus Pauling, who was also working on the same thing, probably would have figured it out in a couple of weeks anyway. It was a lot of people were working toward the same conclusion, and Watson and Crick and those guys discovered it first just by sheer luck. Oh, and this is the Francis Crick Center in the UK. I also took this photo. I was up there. So they've got this giant, beautiful lab named after Francis Crick right in the middle of London, right next to King's Cross Station. So you can go check out your favorite Harry Potter view and then go look at a giant genetics laboratory. So the double helix is the way DNA is put together. Each strand of DNA consists of nucleotide bases connected through covalent bonds. I'm sure you know all about this. The sugar phosphate backbone is where these nucleotides are connected. Each sugar is connected to the opposite side of the next sugar molecule. The positions along this ring are just given numbers, and so you have one attachment point on position 5, and you have one attachment point on position 3. That gives rise to this 5', 3' directionality. Biologically, it's kind of somewhat arbitrary, but DNA synthesis and transcription always runs from the 5' to the 3' end of the DNA. Now, you can have genes coded on either strand, the positive or the negative strand, but it just means that if you're copying mRNA from the positive strand, you move this direction, and if you're copying it from the minus strand, you move in this direction. So there's a directionality, and the actual what determines the positive or negative strand is completely arbitrary, but you have about half your genes are coded on the positive strand and half are coded on the negative strand. Base pairing connects the two strands of DNA in a zipper-like way. It's hydrogen bonding, meaning that it's easily reversed, but when you combine all of these hydrogen bonds together across the entire length of your DNA molecule, that's quite a strong bond when you put them all together. The AT pairs have 2 hydrogen bonds, like right here, and the GC have 3 hydrogen bonds. And that makes AT pairs less strong than the GC pairs, meaning that if you have an area of DNA with a lot of Gs and Cs, that area tends to be more difficult to separate. And that's relevant for designing things like PCR assays and other things that require the DNA to be melted apart and re-annealed. As I mentioned, genes can be coded on either the forward or reverse strand. Forward is the plus strand, reverse is the negative strand. Again, biologically arbitrary, just what we named them. If the gene is coded on the forward strand, it means that the sequence on that strand matches the sequence of the mRNA, which means that actually the mRNA is transcribed from the reverse strand. Very confusing. Again, biologically not too important because it's sort of an arbitrary distinction that we made as people studying it. DNA is wound into the famous double helix shape. We're going to learn more about how DNA is wound up and organized in later lectures, but we all know this, right? It's just a starting point. There's a much more complicated three-dimensional shape that DNA assumes. It's a right-handed helix, so it has the same kind of directionality as like a regular right-hand threaded screw. So if you take like a wood screw or a machine screw and you look at the way the threads are, that's the way the threads should look in a picture of DNA. So occasionally you will find, and you can be on the lookout for this, left-handed DNA where the DNA is actually rendered in the reverse direction. That doesn't exist in nature. I think it probably could if you were to synthesize it, but it doesn't exist naturally. So these left-handed DNA is kind of a funny thing that sometimes you'll see in even textbooks and promotional material. Now you have meiosis as a source of genetic variation. And you can see I'm kind of breezing through a couple of different like bullet points for genetics. I'm not trying to give you an all-inclusive course on it. I'm assuming you probably know most of this stuff, at least to some extent. As we go through the material, I'll bring back elements that I think are worth focusing on and review them as needed for each individual topic. I just want to give you kind of a quick greatest hits of the genetics world. So normal somatic cells have homologous chromosomes in pairs. They're diploid cells, of course. But gametes must be haploid, meaning they must have only one set of chromosomes. The sexual reproduction, if you didn't have that, would be an endless doubling of chromosome count, and it would lead to no real meaningful genetic diversity. You have to cut things in half so that you can combine mom and dad's DNA together and have another new diploid genome. And so meiosis produces these haploid gametes with novel combinations of parental cells. And there's a whole kind of thing with that. You can look at the different phases and, prophase 1, prophase 2, all this kind of stuff. It's a whole thing. We're not really going to worry too much about that. What matters most, and this will come up in detail later on, is the aspect of crossing over. This is taking place during prophase 1, and essentially what happens is homologous chromosomes, so chromosome 1 from mom and chromosome 1 from dad, will pair up, or synapse. Synapse just means like a connection, not it's not only neurons that can synapse, to form a synaptomale complex. And these pairs of chromatids will be broken up and restitched together in homologous or matching areas by the DNA repair enzymes. So what you actually end up with is not just mom's chromosome or dad's chromosome. You end up with this kind of like mixture of pieces from both that are randomly recombined into new things. This schematic kind of shows you what you get. So traits are inherited in this kind of patchwork way. You might get some piece of something from mom, you might get some other piece of something from dad, and the ultimate combination is unique to you. The odds of having the same crossing over take place the same exact way twice is almost impossible. That's why identical twins are such a big deal, right? They're the only people really who are genetically identical to a large extent, because the normal mechanisms of meiosis don't allow for that. So the way crossing over works and the way that genes, like homologous areas, recombine with each other is a fairly interesting topic that's quite relevant for understanding how the broader genetic structure of diseases and so forth. But we'll get into that later. It's not, we're just kind of starting out now. But yeah, crossing over produces novel combinations of genes. So by mixing and matching corresponding regions, meiosis can produce a nearly limitless variety of new combinations. And this is the beauty of life. You can get all kinds of crazy new things that never existed before on Earth. You can have your mom's eyes and your dad's nose and whatever else. Typically, for polygenic traits, you end up with kind of a mixture. You don't just get all of one thing. You end up with kind of a mixture of both. And this is this is the reason really why people would have assumed that genetic inheritance was blended, not discrete in the way that Mendel described. Okay, so I've reached the end here. I think one thing I'm going to have to do is put a marker on the PowerPoint telling me what slide I'm on, because I kind of forgot how many I had to show you today. But that's all I have for you today. Like I said, it's just a quick introduction. If you're new, if you're not familiar with genetics and if you need to brush up a little bit, what I would recommend doing is reading through the textbook or reading through your old textbooks if you happen to have them. Or there's a lot of good resources on YouTube. A channel I've discovered recently called the Amoeba Sisters is quite good. They're kind of fun cartoons that explain biology in a interesting and intuitive way. But yeah, there's a lot of resources online. I think for this course, my goal is to, once we get through some of the preliminary stuff, is to teach you what you need to know on the level of every individual trait and disease. So I'm going to sort of treat each trait or disease as an occasion to give you a little vignette showing you, it's kind of sort of didactic in the sense that it teaches you a little lesson about each trait and how it was discovered. So in one place, we'll really try to focus on linkage and how we determine that. Another place we'll really try to focus on DNA methylation and how that is relevant to things, and kind of work through step by step to kind of weave the learning about genomics and genetics and epigenomics in with the actual stuff you're here to learn about, which is the brain. So hopefully that'll be enjoyable to you. Let's just pop into the chat. Kiana M gives this little smiley, weird looking thing. I don't know what that is, but you're feel free to put anything in the chat you like. I want this to be fairly casual, so you can press F to pay respects or put in whatever kind of thing you like, as long as it's not bad or profane. Hi Harry, could you give us advice on how to be successful in this course? Sure, of course. And sorry, by the way, I realize there's a bit of a delay here. I think there's a few seconds of delay. So Natalie asks, how could we be successful in this course? I think, well, I've never taught the course before, so I don't know what it is that makes people successful. But I think that carefully studying what I say and the material is going to be key. It's not, I think... Yeah, I mean, the normal things that make you successful, I think, would apply here, right? You want regular lecture attendance. You should watch lectures live if you can manage, and certainly view them again in the replay. You can go through and check over parts that didn't make a lot of sense and that type of thing. Do the assignments early and make sure that you have them in on time and you understand them. Talk to your TAs for help with the assignments to make sure that you're doing them right. The TAs will be able to help you with that, as will I if you need it. And ask questions too. I think there are some things that maybe on the first go around, the way I explain it won't really make sense for you, or it may not kind of click. And if you need help, either ask me on office hours, or you can make a meeting with me, or just ask in the middle of class. You just type something into the chat window and I'll answer it, to try explaining something a different way. And that actually can help as well. Sometimes it's just the wording that really is confusing. So yeah, just put in your effort and make sure that you're making an honest attempt to come to class and do the work really well. Once we have the first midterm, once you take a look at the assignments, I guess, and once you have the first midterm, you'll get a sense kind of more about how I run this place and what kind of questions I ask and what I focus on. Okay, so Justin asks, are the midterms and final going to have questions relating to the assignments at all, or just the course material? That's a good question. So no, there's no plan to have anything about the assignments on the midterms. The assignments are sort of self-contained. You'll see that they're very much about following, kind of a set of steps through using a web tool. So they're self-contained. I'm not going to ask you what chromosome is the ACE2 gene located on or something like that. It's not, that's not going to be happening. I guess maybe there some tiny bit of knowledge that you learn from that might filter into an exam question, but you would never need to go back and study the assignments to understand how to do the exams. The exams will be self-contained, and the exams are just going to be about lecture material too. I'm not going to make you you don't have to read the textbook to know anything for the exam. The textbook's just suggested. All right, folks. So we're gonna shut her down there, and thanks to everyone for attending. I hope you enjoyed the kind of YouTube streaming experience. I hope that was. Taylor asks, I can't remember if you said this or not. But will a midterm be just multiple choice, or will it have short, long answers? Check the syllabus, but yeah, it's going to be a mixture of both. I haven't quite determined what the ratio is going to be. For me as an instructor, it's a lot easier to come up with short answer questions because Multiple choice ones are fairly hard to come up with. They take a lot of work to figure out how to make, convincing distractors. Asking a short answer question is much simpler, and I think it actually tests your knowledge better. So probably a mixture of both. I don't know what the ratio will be. I'll let you know closer to the date. For the midterm quizzes, you said they should last 1.5 hours. Will they have a time limit? Yes, the exams will be timed. Of course, if you happen to have accommodations from the PMC, your time will be adjusted accordingly, but the default time is 90 minutes or 1.5 hours. We'll have to see how many questions that actually results in. It's kind of difficult to predict, I suppose. I want to make it, the challenge with these quizzes, because they're open book, they can't not be open book, right? You're on your own computer. I can't stop you from looking at stuff. The challenge is to make them a little bit time limited so that you can't just literally open up a slide, have it next to your exam, and just type in what the slide says. Like, I want to make it a little tough for you to do that kind of stuff so that you're relying more on your memory. But at the same time, I don't want to make you have to rush and panic and forget everything that you need. So it's a bit of a fine line there. But anyway, that's the plan. Okay, so if there are no more questions, I'm going to shut her down now, folks. Enjoy the ending music, and like I say, if you have any questions, you can come to office hours this week, or you can email me. I guess office hours is actually tomorrow, so I'll send out information on that. I'll send out information on that tomorrow. Okay, well, catch you next time, folks.